Hi everyone. We are on section 6.2. In 6.1 we talked about some of the chemicals and the chemical conditions that are needed for microorganisms to grow and thrive. And so in this section we're going to talk about some of the environmental factors that influence their growth. The first thing I'm going to talk about is temperature. And when we're looking at temperature, some of the vocabulary to use is minimum temperature, maximum temperature, and optimum temperature. Depending on where the organism evolved, under what conditions, it can have a different minimum, maximum, and optimum temperature for growth. And usually where you naturally find those organisms, the temperature ranges that are present, or the temperatures that are the most common, are going to determine what that range is for that organism. So the minimum temperature is the lowest temperature that permits the microbe's continued growth and metabolism. The maximum temperature is the highest temperature at which that growth and metabolism can proceed. And the one that we're probably the most concerned about is the optimum temperature. And this is going to be the temperature at which the growth and metabolism is optimum, where it's going to grow its best. And, a lot of it depends on under what conditions did this organism evolve? What was it exposed to? How were the enzymes formed in response to what temperature range? And there's a bunch of vocabulary words that can describe the temperature ranges for various organisms. You might want to look at that. Food, pathogens, and temperature. But on this graph here we have the rate of growth versus temperature. And I just wanted to remind you that this is temperature scale is in Celsius. So 0 degrees Celsius is freezing. 100 degrees Celsius is boiling. So, and as you can see, there's certain organisms that can grow in temperatures above boiling. So let's look at the different categories here. The first one we have is psychrophile. And psychrophiles are illustrated by this graph. And it's showing you that psychrophiles typically have an optimum temperature, a little bit above freezing. And where you're going to find psychrophiles are in places, for example, in glaciers, where it's always frozen, it's never melted. In areas of the ocean where it's very cold and it's always cold. Because notice, room temperature is closer to around here, our normal room temperature, high 60s, 70, around there. Notice that these psychrophiles, that's above their maximum temperature. So you're not going to be able to a psychrophile isn't even going to be able to exist, let alone grow, in moderate temperatures and what we would call our typical room temperatures. So these are going to be in those very cold temperatures. Psychrophiles are very unlikely, if any, if at all, ever cause disease, because disease has to happen in an organism that's living, and most of those are going to be living in more moderate temperatures. So you don't usually have to worry about getting sick from a psychrophile, and most of us will probably never see a psychrophile in real life. I'm going to skip to mesophiles. Meso means middle. And here we have the mesophiles, minimum temperature, maximum temperature, but look at this optimum temperature is kind of around the high 30s. Hopefully you remember that 37 degrees Celsius is human body temperature. So most of the microorganisms we come in contact day to day are going to be mesophiles. In fact, we're mesophiles. We grow at these moderate temperatures. And so these are the temperatures that are comfortable for most plants and animals. And one of the things that you're going to see is mesophilic microorganisms are going to be the ones that cause disease, because they're going to grow in the same environments that their hosts will. They live at in the same temperature. Most decomposition is going to occur by mesophils. So when your food spoils, it's going to be mesophilic microorganisms that's spoiling that food. Even the microorganisms that are in your refrigerator spoiling your food are going to be mesophils. Remember, our refrigerator temperature is somewhere down here, and it's supposed to stop the growth of mesophiles, but the reality is our refrigerators aren't always that low, and we do get some growth of mesophilic bacteria even in our refrigerator. Some people will say, oh yeah, psychrophilic bacteria, they grow in the cold, so that's what you're going to have in your refrigerator. Remember, those psychrophiles have to be cold all the time. The mesophiles got in your refrigerator from the plants or the, any kind of food that you put in there, and those all came from mesophilic environments. Thermophiles can grow at higher temperatures. Now, if I give you an idea of how hot, like 50 to 60 degrees Celsius, I could put my hand in 50 degrees Celsius water, but I'd pull it out and be like, oh, it's a little bit too hot. Don't want to leave it there. Okay, so that's pretty hot. And when you get up to 70 or 80, that's very hot. You don't want to dunk your hand in there and leave it there for any amount of time. It would be painful. But there are certain groups of microorganisms that are what we call thermophiles, and they can live in these temperatures. And again, notice that their minimum temperature for growth is not even in the area where most mesophiles grow. Thermophiles can be found in places, well, just day to day, if you have a compost pile. And especially when it's starting to get cool out, you might notice when you dig into the compost pile, you might get steam coming out of it. You might notice that. And that's because you'll have microorganisms that are metabolizing so fast that they're generating heat. And they actually kill off some of the mesophiles in the hottest part of the compost pile. And a lot of those will be thermophiles. Areas around very hot water or volcanoes, on the fringes of that, where it's kind of the soil's very warm, might have thermophiles in there. The last group we have here are what we call extreme thermophiles, or you might hear them called hyperthermophiles. And so here, look at this. We have 100 degrees Celsius, boiling water, and these microorganisms are going to grow in that region. These are places like hot springs, like at Yellowstone, where the water is boiling or nearly boiling. There's some geothermal vents in the deep oceans that are really hot that can have these. And so why isn't the water just going away? Why isn't everything boiling away? Because they're also under pressure. So these hot areas under pressure, most of these are in the ocean. So we do have some of these hyperthermophiles or extreme thermophilic organisms. So there's all different organisms. The only ones that are likely to cause any kind of disease or be our normal microbiota or cause food spoilage, things like that, are going to be these mesophiles. Here it gives you a little of information. Here's some psychrophiles growing on a glacier. And these are photosynthetic organisms, but they happen to be red. Talks a little bit about mesophiles and thermophiles. Okay. The other big topic I wanted to talk about in section 6.2 is about oxygen. So the conditions for oxygen really, the presence of oxygen has a big impact on how microorganisms grow. And here it's telling you that atmospheric oxygen can influence microbial growth, but it's not only an important gas for living, but oxygen can also exist in toxic forms. And so what has happened is all the organisms that have evolved to use oxygen in the metabolism had to also evolve enzymes to detoxify oxygen. So oxygen is actually a toxic agent to organisms that don't have the right enzymes to detoxify it. So here it's telling you that microorganisms can fall into three categories: those that use oxygen and can detoxify it. And that's us. We use oxygen, and we also have ways to detoxify the small percent of oxygen that forms into toxic forms. There's some microorganisms that can neither use oxygen nor detoxify it. And we're going to talk about these organisms. They'll be called anaerobic, meaning they don't use oxygen, and oxygen's poison to them. And finally, we have organisms that don't use oxygen in their metabolism, but they can live in the presence of oxygen because they can detoxify it. So what's the scoop on toxic oxygen? Who knew, right? So one of the things that in any organism that's going to live in the presence of oxygen, there's this stuff that naturally forms. And here it's called hydroxy radicals. You might hear these called free radicals. Here they have the OH here, but this is the same. This is equivalent right here. We call these free radicals. Oh, here we go. Superoxide ions. That's what these are. And you can get these also. But here's these superoxide ions. We also call them free radicals. And you know about these because when you're selecting foods to eat, you're told to eat colorful foods. You're told to eat antioxidants. We hear blueberries are really good antioxidants. Colored vegetables, leafy greens, things like that are all good antioxidants. And they say they're good for you. They prevent cancer. They extend your life. They make you healthier. And it's because they neutralize these superoxide ions, these free radicals. And so these are naturally forming as a product of metabolism in tissues of organisms that live in the presence of oxygen. And so fortunately, humans included, organisms living in the presence of oxygen make an enzyme called superoxide dismutase. And you might hear that called SOD. So superoxide dismutase, or SOD, is an enzyme that detoxifies these superoxide ions and produces hydrogen peroxide. Now, hydrogen peroxide you might have in your cabinet at home, but that also is toxic to us. It's not quite as toxic as these superoxide ions, but it still is toxic. So we also have another enzyme, and the enzyme is called catalase. And what catalase does is it takes hydrogen peroxide and converts that to water and oxygen, and those are harmless to us. So every organism that lives in an oxygen-containing environment, humans included, have to have enzymes to detoxify oxygen. And those enzymes in particular are superoxide dismutase, SOD, and catalase. Okay. Now, turns out that there are microorganisms that have some variations on this. Just like humans, microorganisms can have SOD and catalase. And because of that, they can grow, it influences how they grow. So under what conditions? Let's look at this table. First, it's talking about this medium called fluid thioglycolate. Where did they mention that? Here it is, fluid thioglycolate. This is a medium that we use in the laboratory. I'm going to go down here just to show you. It's a medium we use in the laboratory. So here's some test tubes or some screw cap tubes. And in here is this medium. It's semi-solid. It's got some agar in it. It's kind of thick. And what they do, it's got some ingredients in here that help drive out oxygen and keep parts of it oxygen free. So what happens is you autoclave it, you sterilize, you heat it up, and it drives the oxygen away. But oxygen comes back in and from the air, and only the very top will have a high amount of oxygen. So if I looked at this medium, I put a little gradient here. I would say I have high oxygen here, and then I have less and less and less, and I have no oxygen down here. Okay, so this is a medium, fluid thioglycolate, and you're going to see this in lab also, is a medium that you can have high oxygen at the top and less at the bottom. And when you inoculate this, you take some bacteria on kind of a needle, and you stab it all the way down here. So the bacteria are only going to grow where their oxygen conditions match the bacteria. So let's go back and look at this diagram, this picture. We can categorize bacteria into several different groups depending upon their oxygen uses. And first we have aerobes. Now, we are aerobes. We use oxygen for our metabolism, and we have enzymes to degrade toxic oxygen products. So if you look at the fluid thioglycolate medium, the bacteria, the aerobic bacteria, are just going to grow at the very top where there's oxygen. And it says many different kinds of fungi, protozoa, bacteria are going to be aerobes. You might hear this called obligate aerobes. They have to have oxygen. Look at this word, microaerophile. Micro means a little, aero means oxygen, phile means loving. It likes a little bit of oxygen. So it says they are harmed by normal atmospheric oxygen, and that's because they have actually a little bit lower amount of SOD and or catalase, but they have some, and they need some oxygen for metabolism. So they like a little bit of oxygen. So if right up here is the highest amount of oxygen, they can't grow there because it's too toxic. But right down here where the oxygen gets a little bit lower, they're going to be able to grow. They need oxygen to grow, so they're not going to grow in this anaerobic region of the tube. Microaerophiles. And things like Helicobacter pylori, that organism in our stomach that can cause stomach ulcers, Borrelia burgdorferi, causes Lyme disease, they're both microaerophiles. Facultative anaerobe. Facultative is a word that says they can, but they don't have to. Anaerobe, let's see, an aerobe is oxygen loving, and means not. So an anaerobe is going to grow without oxygen. So a facultative anaerobe can grow anaerobically, but it doesn't have to. So it can grow aerobically or anaerobically. What we have right here is the facultative anaerobe is actually going to grow throughout the thioglycolate medium because it can grow with oxygen or without. The trick is when you're growing aerobically with oxygen, you grow faster. So here we have a facultative anaerobe growing faster at the top and slower at the bottom, but can grow throughout the whole tube. And we've got a lot of facultative anaerobes associated with humans. It says many gram-negative intestinal bacteria. Staphylococcus, which we find a lot on our skin, they're both facultative anaerobes. An anaerobe is one that can't grow in the presence of oxygen. So, and that's because it doesn't use oxygen, but it also does not have SOD or catalase. So oxygen is toxic to it. And so it's only going to grow here where there's no oxygen. And again, we have bacteria in our mouths, we have bacteria in our intestines that are anaerobes. Finally, aerotolerant anaerobe. It tolerates oxygen because it has some SOD and or catalase. But it doesn't use the oxygen, so it's an anaerobe. So, so it's tolerating the oxygen. It's totally neutral about it, and it's going to grow throughout the tube. So it doesn't matter if there's oxygen there, because it's not toxic to oxygen's not toxic to it, but it doesn't use oxygen, so it can grow anywhere in that tube. And that's going to be lactobacilli, streptococci. Some of these are bacteria that we use to make things like yogurt. Here's another diagram that shows the same thing, but kind of in real life. So here's the 4 different thioglycolate tubes. Notice where it's clear the bacteria are not growing. So there's no bacteria growing here, but there is some growing there. And so this first one is an aerobic organism. The bacteria are just growing at the top where there's oxygen. This one is a facultative anaerobe. It can grow with or without oxygen. It's cloudy in both areas. But with oxygen, it's going to grow faster. This one's also a facultative anaerobe. It's a little bit harder to see. You can see where it is cloudier up here. So it is growing a little bit faster up there than down here. And then this one is an anaerobe where it's not growing where there's oxygen, but it's growing where there's less oxygen. Let's see. The last thing I wanted to talk about in this video was, oh, actually I have pH. No, actually I think I'm going to stop right there and start a new one.